Yeah, we could probably go ahead and get started. So I'll maybe start with some course logistics. So we're in the middle of week 2, which means lab 2 has been out. So hopefully everyone's been making progress on that. One thing that I don't think I was able to show you before, or I don't think we had yet, was some folks in my course staff were mentioning that it might be nice if we just organized all the different tools that you're going to be using throughout the quarter, as well as a bunch of other bioinformatics tools that might be helpful. So I've added this curated list of just a bunch of bioinformatics tools, and I've kind of broken them down by just general category of task that each tool does. So things like how do we assess the quality of a sequencing experiment? How do we do genome assembly? And then these other things you don't have to worry about yet, but we'll talk about them a bit later. So hopefully throughout this course and then also in the future, hopefully this will be a nice resource of just like, here's a bunch of tools that you could look into if you're ever interested. The other thing I want to mention is I do want to again remind folks that I have a link to an online Python textbook that I wrote. So if there's anything about like Python data structures that you want to review, so things like how do I use a hash table in Python or like a hash map in Python or whatever. So this textbook, maybe I'll highlight the relevant sections. I think they're going to be useful for this lab for like counting the unique k-mers and stuff. If you go to the week 4 section, I have an overview of different data structures. I think 12 is a prereq for this class, right? Okay, so everyone's like learn these data structures in theory. So the Python list is an array list. Python tuple is like effectively a multi-value type array, essentially, like a fixed length array list. So I guess just an array. A set is just a hash table, and a dictionary is just a hash map. So definitely, if you think like, oh, I wish I had access to one of those data structures I learned about in CSE 12 to make things faster, these should hopefully be good references for that. Cool. So yeah, so lab 2 is out as always. Hopefully folks are working on it. Come to lab section if you want to get help from one of the members of the core staff. And yeah, any logistical questions from folks before I jump into today's topic? Questions, concerns, comments, dreams, aspirations? No? No? Okay, cool. So let's pick up exactly where we left off. So hopefully everyone can see my screen. Let's see. So where we left off, we basically came up with, and I'll just kind of like briefly summarize the most recent slides. So we used this De Bruijn graph genome assembly approach to be able to reconstruct our genome sequence from those small fragments. I mentioned in practice, there's some real world challenges that we might have to face. Things like you might have low quality base calls in the reads that you might need to do a quality assessment to just, or do I have to rerun the sequencing experiment from scratch? Things like maybe the overall sequencing experiment is fine, but maybe some of the bases at the ends of the reads are bad quality, so maybe we got to trim those. We talked about things like even if we only keep the high quality bases, there's still going to be inevitable errors. So there's things like bubbling that I briefly mentioned that you don't have to worry about. But basically, the De Bruijn graph can get kind of messed up if you have sequencing errors and other real world imperfections that it has a lot of really clever techniques to overcome. So for the purposes of this class, the key takeaway is Spades and other genome assemblers are really fancy. Just use them off the shelf. They handle all these weird situations. And if you ever go to grad school and want to build an assembler yourself, then you'll have to deal with those real world imperfections yourself. But for our purposes, we're just going to assume that these tools do those well enough. And then we mentioned we can use tools like Quast to calculate all of these different metrics like N50, NG50, create plots like this where the area under the curve is effectively a measure of how good our assembly is. And we ended by saying, okay, now we have this genome assembly. I can then use BLAST, which is basically like the Google search of DNA sequences to see what things have I seen before that this looks like. So without any virology expertise, we as bioinformaticians can figure out, hey, this unknown pathogen matches most similarly to this bat SARS-like coronavirus. It has like 95% coverage and like 89 coverage and like 89% identical in that 90% of the genome. It's next closest to SARS, but more distant from SARS than these bat-like ones. So maybe these are close in between sequences, like maybe some intermediate species that got infected or something. And what this tells us is now we can hand this off to a virologist and they could do some type of real world intervention of like, hey, maybe the therapeutics that we used during the SARS epidemic could be repurposed or slightly modified for this new pathogen It's a coronavirus, so that tells us things about how it functions. So maybe that'll lead to some like downstream virology research. Basically, a lot of information is helpful just from this sequence. Okay, so questions about anything we talked about last time? Any concerns? Anything that you want me to clarify? Anyone on Zoom, feel free to type in the chat. Feel free to raise your hand if you want to unmute. Folks are all good. Awesome. Okay, so let's kind of summarize what we finished. Last time, we assembled our genome. Perfect, all done. Should we call it a day? We finished this class. All good? World is saved? Yeah? No? You're saying no? Why not? Other than my snarky tone.
It's like a bunch of A, T, B's, and C's.
Yeah, it's the genome.
So we should do something with it. Like
So here you go.
some sort of analysis.
So what's missing here? Like why is this? So you said it's just A's, G's, and T's. So what's the problem with this?
Like all the information, but what are you gonna do with it?
Was it all the information? What information do you see?
Or like it's so general you don't really know what to do.
Okay, don't really know what to do. And you had your hand up, or? Well, this is the first position. This is the second position. This is the third position. The problem solved. I get a Nobel Prize. No? Okay, yeah, go for it. Go for it.
Could you now try to find like the amino acid sequence?
Ooh, so you said find the amino acid sequence. So basically, what's your critique then? So you have a critique that you have now come up with a potential solution. But your critique, I'm gonna just kind of like paraphrase what you're implying is, I don't know what this means. Like it's just a bunch of A's, G's, and T's. Like I'm not a human. I'm a human. Whoops. Uh-oh. I've been outed. I'm an alien secretly. I'm a human. I don't know what these A, C's, G's, and T's mean, right? I have to figure out some way of coming up with some information from them. Things like maybe figuring out what are the positions of the genes. Things like, oh, hey, if I find the positions of the genes, what proteins do they encode for? What are the different functional units of this genome? There's all this downstream information that just knowing the letters doesn't really tell me. So the next section we're going to talk about is going to be about finding genes. And to be able to find genes, we have to first kind of do a little bit of a recap about the central dogma of molecular biology, which is going to be our like signal that we're going to use to find the genes. So first, let's do some bio review. Since this is a CSE class, and I'm assuming some folks might have taken bio quite a while ago. So what is the central dogma of molecular biology? Anyone remember? Any thoughts? Yeah, go for it.
DNA transcribes to RNA, translates to protein.
How did you guess? Wow, exactly. Perfect. So yeah, DNA gets transcribed to RNA. RNA, and specifically mRNA, gets translated into protein. So the central dogma of molecular biology, it's kind of describing this forward in motion kind of passing of information. In reality, there's more steps. You can have DNA self-replicate, which is kind of weird. You have like DNA replication. You can have RNA reverse transcribed to DNA. You can have RNA that's functional on its own. You can have proteins that do weird stuff. In reality, it gets much more complicated like everything in biology, but for our purposes for today, this is it. DNA is kind of the wealth of information. That information gets transcribed into RNA, which is kind of like the copy of that information that can go out and do stuff. And then protein takes that copy from the RNA and then creates we create the protein to actually do the functional stuff. So let me just kind of summarize each step one by one. Transcription, DNA to RNA. You should kind of imagine, actually, maybe let me go back to the previous slide. So where does DNA exist in a cell? Like why does it exist? It's in the nucleus, right? And for our simplicity of today, we're going to assume protein is the functional unit of the cell. Protein is doing stuff outside of the nucleus. How do I get proteins out there? So you should think in terms of our like very simple analogy that DNA is kind of like a library that has instructions on how to make every single possible thing I might ever want to create. But I can't take things outside of, like I can't take the DNA out of the library. It has to be fixed inside the library. But I can make a photocopy. So you should imagine that RNA is kind of like a photocopy of the specific thing that I want to build. So I can't take the instructions themselves out with me, but I could create a photocopy of what I want to build and take that photocopy out with me. So the RNA is able to leave the nucleus. And then protein is that actual thing that I want to build. So given the instructions that I copied, I can then take it elsewhere in the cell. I think the ribosome probably. This is where I suck at cell biology, but I take this mRNA and I use it to create the protein through a process called translation. So that's kind of why these different pieces matter. For our purposes, though, we're computer scientists. We're going to pretend we don't care about those things. How can we summarize these two processes from our perspective in this class? We can imagine a DNA sequence is just a string over the alphabet ACGT. We can think of RNA as just being a string over the alphabet ACGU. And for the simplistic purposes of this class, we're going to assume the high-level summary of transcription is just take all the T's and replace them with U's. So imagine I have this DNA sequence, and I can imagine transcribing this DNA sequence is just literally taking every single T, replacing it with the U, and copying everything else exactly as is. Okay. Questions? Everyone's good? Okay. Translation is a little bit trickier. So translation, it's the process by which mRNA gets turned into, not turned, but like basically we synthesize protein from an mRNA sequence. So with DNA, every letter in the DNA sequence maps to a specific letter in the RNA sequence. A to A, C to C, G to G, T, map to U. But with proteins, we have 20 amino acids. So we should think of a protein sequence as kind of being a string on this alphabet of 20 amino acids. And we have some way of mapping from RNA to these amino acids. So from ACGU to these 20 amino acids. And how do we do that mapping? It's something called the codon table, where a codon is a 3-letter sequence of RNA. And each 3-letter sequence, each codon, maps to a specific amino acid. So for example, the most common start codon, which is what we're going to be assuming in this class, is always going to be the start codon. In real biology, it gets messier, but for our purposes, we're going to assume always start codon is AUG. But this codon encodes for an amino acid called methionine, which is going to be represented as the letter M in the amino acid alphabet. We could take any arbitrary codon that we want, so like maybe CGA, first letter is C, second letter is G, third letter is A. CGA codes for arginine, which we're representing as the letter R. So basically each 3-letter chunk, I can look it up in this table, see what is the amino acid it codes for. There's 3 special codons that don't do that. What are those codons? Yeah, go for it.
I know one of them is ATG.
Ooh, ATG. So ATG actually does code for an amino acid. It's the start. ATG is AUG, so it codes for methionine. It is special though because it's the start codon. But any other ones? Yeah, go for it. Exactly. So UGA, UAG, and UAA, these 3 are called the stop codons. These are basically what tell the translation machinery, stop translating. This is the end. They don't code for an actual amino acid. So the start codon actually codes for methionine. Again, simplistically for this class, in other contexts, there might be other start codons, but for our purposes, the start codon is AUG and it codes for methionine, but the stop codons don't code for anything. They just say, cut off right here, we're not translating anymore. And the algorithm that we're going to be describing to summarize translation in our computer science minds today is start with a start codon that's relatively early on in the sequence. I think in the lab, I have you do it on a problem where we're assuming it's the first one. In practice, there might be other AUGs that happen also in this mRNA fragment that are in what's called the 5' untranslated region, 5' UTR. That's the 5' region. So DNA goes 5' to 3' start to end. The 5' UTR, 5' untranslated region, is the part of the RNA at the beginning that is untranslated, doesn't get translated. So the start codon is going to be a relatively early on AUG. And then starting with that start codon, we go 3 letters at a time, encoding each amino acid. So until we hit a stop codon. So let's try it with that same RNA sequence that we had created. And let's start translating this. So I'm looking, and this looks like the first AUG, which for this example, I'm going to assume the first AUG is the start codon. Again, the first AUG might show up in your 5' UTR, in which case there's fancier ways that you would have to figure it out. But for our purposes, let's just say it's the first one. And if I look it up in the codon table, AUG codes for M. The next codon, so I just slide 3 letters over. The next codon is GCU. If I look it up in the codon table, this codes for A. Slide over. The next one codes for T. The next one codes for T. The next one codes for H. The next one codes for I. The next one codes for A. The next one codes for S. So I'm just looking each of these up in the codon table. And then the next one that I hit, UGA, I look it up in the codon table. That means stop. Translation is done. So this mRNA sequence encodes this protein sequence, where these are all amino acids. And we don't include the stop codon in our protein sequences. It doesn't actually code for anything. Awesome. Questions? Kind of makes sense. Okay, so how is this helpful? I just went on this tangent to like remind us of some molecular biology. How is this going to help us find genes? That was our original problem. Well, imagine that I've assembled this DNA sequence. Let's say that it's a single sequence from 5' to 3'. Again, you don't have to worry about what the 5' and 3' mean. Just remember 5' is start, 3' is stop, or like beginning, end, I should say. 5' is the beginning, 3' is the end. So this fragment gets read out in this direction. And remember that DNA is double-stranded, where the other strand is the reverse complement. So the other strand, if I only assembled this sequence, the other strand, I could just reverse and complement it. And the second strand gets read backwards. It goes from right to left. 5' to 3'. Okay, so what can I do here? What I could do is I could transcribe, I'm going to call this the forward strand, and I'm going to call this bottom one the reverse strand. Which strand you define as forward versus reverse is arbitrary because DNA is double-stranded, so it's kind of meaningless. Usually we call the forward strand the one that is where all the genes are encoded. But sometimes genes are encoded on the reverse strand, so even then it's kind of meh. So either way, but I'm just going to say the one that reads from left to right is the forward strand. The one that reads from right to left is the reverse strand. I can transcribe both of those, again, just replacing all the T's with U's. And then what I could do is I could try to brute force every possible set of translating that I can do. So with the codons, I had to pick a starting point. And then from that starting point, there's no questions asked. I just look up that amino acid, like look up this codon in the codon table, look up the next codon, next codon, next codon, next codon, one by one. So if I pick a starting point, for example, if I start here at the first G in the forward strand transcribed RNA, the first codon is GUG, which I could look up in the codon table, and it codes for this amino acid. Next one is AAA, codes for this amino acid. Next one is CUU, which codes for this amino acid, etc. So if I pick the first letter and do translation starting with all the codons starting with that letter, I can come up with every single possible amino acid translation. And then what I could say is, well, I'm not sure if this is where translation starts. What if translation actually starts in this letter? So my codons, they're 3, 3, 3, 3. What if that first letter was shifted by 1 and then do 3, 3, 3, 3, 3, 3? So if I started translation here, well, UGA, that's a stop codon. AAC, that's this amino acid. UUU, that's this amino acid. And I kind of start doing every possible translation starting from the second letter. And then I could do the same exact thing starting from the third letter. So what if it's not the first letter that I'm starting with, not the second letter? What if I'm starting with the third letter and then going one by one by one by one by one? So GAA codes for this amino acid, ACU codes for this amino acid, and kind of do all of those translations. So in other words, let me brute force every single translation on this strand. Okay, so I'm going to pause and ask a clarifying question. So I did starting from this letter, I did translation starting from this letter, and I did translation starting from this letter. Why don't I have to also do translation starting from this letter? Maybe someone new, but I'll come back to you. Oh yeah, go for it, go for it.
Because basically, because you've done sections of 3, starting at that A is just, it's basically taking like the first one you did, where you started at that G, because it's every three, so then at the next, like the fourth one, that's just gonna be the sequence that you did for the first one, again, but without the first codon.
Exactly. Perfect. So the answer was, well, if I do it codon by codon by codon, this letter, if I started translation on the fourth letter, this is the exact same amino acid sequence that I'm going to get as when I started at the first letter, but just chopping off that first codon. So actually, the only possible translations that I could get are starting at the first letter, the second letter, the third letter. Every subsequent letter is just one of those three, but just skipping some of the first letters. Okay, so I'm going to introduce another terminology. We call these open reading frames. So a reading frame, and you might have heard of like frame shift mutations in genetics or something like that. A reading frame is one of those three-letter window starting points. So starting from this position is one reading frame. Starting from this position is one reading frame. Starting from this position is one reading frame. And then starting from the fourth letter is the exact same reading frame as when we had started from the first position. So there's only 3 reading frames on this strand. Those are the frames that I could start my translation from. Okay, folks with me? Kind of makes sense? Okay, well, I brute forced that on the forward strand. Why shouldn't I just do that on the reverse strand? So I could do the exact same thing on the reverse strand. I could do, let me try translating starting with this letter. So AUA as the first codon, then UUG as the next codon, then AUU as the next codon. Again, it reads from 5' to 3', so the reverse strand reads from right to left. But yeah, I could start translation here. This is one reading frame. Then I could start translation here. So UAU, UGA, UUA, etc., starting from here. And then I could start from the third reading frame, AUU, GAU, etc. And now I get 6 what we call reading frames. I get 3 on the forward strand, 3 on the reverse strand. And every single protein that I'm encoding in my genome must have been encoded in one of those 6 reading frames, just by definition. Okay. Kind of makes sense. It's getting a little bit weird, but hopefully folks are still with me. Okay, so what can I do? Well, let me just try all those brute forces, and let's imagine that this is one of those outputs. So the output of one of those brute force translations starting at this position will get me a protein sequence that looks something like this, where I'm just using $signs to represent stop codon. And the question is, do any of these, so I have a couple questions for myself. I have a question of, does this open reading frame contain a gene, or does it have multiple genes? Like, what genes does it have? And where do those genes start? So they might not start at the very beginning of one of those little windows. I know every single protein that's being encoded here has to be within 2 of these stop codons, right? It has to be somewhere in between each of these, because I can't have a stop codon in the middle of a protein. So which of these windows codes for proteins? And like, where does the gene start there? And ideally, some additional information that I want to be able to discover is, what are those genes? So like, for example, if I say this is a gene and that's a gene, what do they do? So maybe like this one's collagen, and maybe this is part of the ribosome. So that's kind of my end goal, is what are the genes and like, what do they do? Okay, so is the problem kind of clear? We have this unannotated now protein sequence, but it has like the stop codons embedded throughout. And I want to see which of those windows are proteins, like real proteins, and what do they do? Okay. So what ideas do people have? So if I presented you with this problem and I asked you, go solve this as a project, what are some sources of information you might use? Any thoughts? And the theory of evolution is going to come in here. So remember, today is 2025. So we're not the first people to look for genes, and we're not the first people to ever discover genes. So we have hundreds of years of biology at our disposal. I'll come to you second, maybe you first and then you second. Yeah. I could compare them with BLAST, so that's one idea. I could take each of these and then look on BLAST, and then what would I do with that? Awesome, awesome, perfect. And then what were you gonna say?
Like you have like a reference source of like different genes,
Mm-hmm.
and you put in like the protein sequence that you get and see which like genes are the most similar to the protein like, protocol.
Perfect. So I'm going to merge those two answers. So the summary was basically, hold up, this is 2025. We have like hundreds of years of biology at our disposal. People have been looking for genes for centuries, maybe not centuries, maybe like a century or so. But we have these massive databases of genes and like protein sequences, DNA sequences, RNA sequences. What if I take all of these and look them up on those databases, maybe using BLAST, maybe using them other techniques, and see what things do we already know about that look kind of like these. And that is exactly what people typically do in practice. So here is a pipeline known as Proca. You're not actually going to be running it. I don't actually, do we have them run Proca? I forgot. I think I had trouble getting it set up. Yeah, so I think I omitted this from the lab just because it was a pain to set up or something like that. Maybe I did set it up. I don't remember what the problem was. Either way, in the real world, you'll probably use this at some point if you're ever doing bacterial genomics or something like that. But basically, the Proca pipeline, this is a pipeline that uses a bunch of different tools that do exactly this. They take databases that we already know about, scan all of the things that we just produced, so the DNA sequence that we assembled, the RNA transcripts that we can transcribe from that DNA, and the 6 different reading frame protein assemblies, or like protein translations, I should say, not assemblies, protein translations that we've come up with, and query them across all those different databases. So just to kind of give you a roadmap of what these things mean, the cylinders are databases. So PFAM is a database of protein families. SwissProt is a database of protein sequences. If you have a user protein sequence database, like you are an expert in some specific topic and you have your own custom one that's not like a third-party one, you could also provide that here. So BLAST, you could run it on your database and SwissProt to do protein annotation of like, I think these are real proteins. Hammer, so I think a couple people mentioned they've taken Pavel's class. Anyone taken like the hidden Markov model class that I think the CSE department has? I think it's like CSE 151 or something, or 150A or either way, no worries. So a hidden Markov model is one type of probabilistic model. If we're trying to be fancy and like market it to people, we could say it's machine learning, even though it's just statistics. But either way, so Hammer is a tool that uses these hidden Markov models to try to infer like matching against a given family of known sequences. So it basically will construct this hidden Markov model from the different protein families that you have, as well as I think Tiger is some type of an RNA sequence database, but it'll take families of proteins. Maybe these are all, have people taken cell bio? Probably not, but yeah, you've taken it. Okay, so do you know what a GCPR is? G coupled. Yeah, so what is that? Oh, that's okay. You know what it is though. That's good. So for example, let's imagine, are there markers in here? I think this whole thing's a whiteboard. Oh, actually the people on Zoom won't be able to see it, so I'll just draw on here. So for example, let's imagine here is my beautiful, oh, actually I have a tablet. Oh ho. Modern problems require modern solutions. Let's draw. Okay, so I'm gonna do like an example of what I mean by protein families. So in general, and I'll just kind of talk while I'm setting this up. So in general, the structure of a protein has strong implications on what that protein does, because really proteins are just these like things that have chemical and physical properties that are just like floating in the cell, and then they hit something and they're like, oh, hey, this feels nice. And then that causes some cascading effect. So let's say that this is, and you're going to see that in another life, I probably could have been an artist if I wasn't a computer science. Why is my thingy not going to that screen? How do I tablet go to that screen? Let's see. Tablet properties. This is like cramping my style. I was like ready to show off. Mapping. I want to go to, how do I go to the other screen? Screen monitor 2. Okay. Oh, nice. Okay. So let's say that this is a cell. I'm going to draw the cell looking like this. Oh, geez. So why did I draw 2 lines here around the cell? Ooh, cell wall. Let's assume, yeah, the good question. Let's assume this is eukaryotic. I guess plants are also. Let's say this is an animal cell. But same concept. So what do animal cells have? Ooh, so perfect. So the phospholipid bilayer, exactly. So your cell membrane is what's called a phospholipid bilayer. Let's like zoom in onto this segment. So let's say like this is like over here. I'm zooming in over here. So this is like my window that I'm zooming in on. Actually, I don't feel like drawing a box around it. So let's say that I have my phospholipid bilayer. What does that mean? I have phosphate groups on the outside. They have lipids or fatty chains on the insides. And the cell membrane is just a bunch of these kind of next to each other, where these little circles are the phosphates, the chains in between are the fats, and it's a bilayer. There's 2 layers of phosphates where the inside is fat, the outside is the phosphates. So is the inside of this hydrophilic or hydrophobic? Ooh. Hydrophobic. Yeah, so they're fat. So think like when you pour oil in water, oil separates from water because fats are hydrophobic. So this middle part is hydrophobic. Hydrophobic. And then the outside and inside are hydrophilic. So now going back. So what's a GPCR? G protein coupled receptor, I think. Swivel. Okay, perfect. Perfect. So I mean that exactly. So basically, so you said it's a membrane bound protein. What does that mean? It's a protein that looks something like this. I mean, this is not drawn to scale, but it's like a protein that looks like this, where it kind of like squiggles in and out of the membrane, and it has some receptor here. Let's say that my receptor looks like this, where if something floats along that kind of like fits into that receptor, it's like, oh, hey, this feels nice, awesome. And then that causes a cascading effect that then does something in the cell that then does something. Then we give it to the cell biologist and they know what they're doing. So let's think about what this protein looks like. So this protein, if I were to take the tips of it and kind of like stretch it out, well, what does this part look like? Well, this is, and remember the amino acids have chemical properties. So this is an amino acid that's hydrophilic. Oh, now I'm inside the membrane, hydrophobic. Oh, now I'm back outside, hydrophilic, blah, blah, blah. So the contents, and basically in this receptor protein, the contents of these are not super functional. The only thing that really matters is it hydrophilic or is it hydrophobic? So that it's if it's inside the cell or it's outside the cell. Oh, sorry, if it's inside the cell membrane or outside or inside the cell. But the actual like item, like the actual amino acids that you pick don't really matter too much. It's just kind of an anchor to hold the protein in the membrane. But like these amino acids over here matter a lot. These are the amino acids that are going to be like forming the 3-dimensional structure that binds to whatever this molecule is. And then also what's on this side matters a lot because this is what's going to be triggering the cascading effect inside the cell. So there's kind of a few different domains that we're describing here. We're describing these hydrophobic domains, hydrophilic domain, hydrophobic, hydrophilic, and also this like whatever this molecule is binding domain. Or it could be like a photoreceptor if it reacts to light, or it could be some other thing where the shape of that impacts it. So what I'm getting at is Hammer is able to come up with probabilistic models that summarize protein families and even like domains of protein families. So I could have like a class of proteins that are photoreceptors, but they could be doing very different things, but they all are going to look kind of similar at this part. And Hammer might be able to predict, oh, hey, this end of your protein looks kind of like the photorecepting part. So it might say, oh, maybe this new protein that you have is some type of a photoreceptor. Maybe it has this like hydrophilic, hydrophobic, hydrophilic, hydrophobic, hydrophilic alternating pattern. So maybe it's a transmembrane protein, and it can try to predict based on those features that it's learned from this big database of proteins, what is this protein's function? There's a couple other things. I'll just kind of like run through these quickly. I think we do run Prodigal, right? Yeah, so Prodigal is one of the tools that I'm having you run. It's part of the Proca pipeline, but it is, all these are like the green things are standalone tools. So Prodigal, you give it the coding sequence, like the actual just DNA, I think. And then, yeah, I believe you just give it the DNA assembly, and then it outputs, no, yeah, so Prodigal, you give it the whole assembly, and then it outputs what it thinks are like the DNA coding sequences that then you could feed to any of these, or you can feed it to the signal P thing. That's yet another tool that does something about peptide signal prediction. I don't really know what that means. I can predict non-coding RNAs. I could predict rRNAs. I could predict tRNAs. But basically, my takeaway here is all of these tools are leveraging big databases of all these existing proteins, RNA sequences, DNA sequences that we already know about, and just looking up using various string matching techniques like BLAST, probabilistic techniques like Hammer, using these different techniques to see what looks similar. Okay, does that rant kind of make sense? I went on a somewhat lengthy tangent, as I sometimes do. Okay, kind of make sense? Awesome. Okay, go for it, yeah.
So just to recap, it's to make sure or look at what's similar, but not like an exact match. So you can kind of make a guess of what it does.
Exactly. So the question was like, is it just looking at what's similar, but not necessarily an exact match? It could be an exact match. So let's say that this unknown pathogen actually was just SARS. Then I've already seen SARS. I know what proteins are in SARS. So when I do things like matching to BLAST database or like matching to Hammer or something, it might actually perfectly match a protein that I already have seen, or it might not, in which case it'll output what it looks similar to. So however much it can infer based on what it already knows, which it can infer more, the more similar it looks like to things I've already seen. It'll be a little bit fuzzier, the less similar it is to things I've already seen. Yeah, great insight.
Thank you.
Yeah, no problem. Yeah, great question. Awesome. Any other questions? Okay, so let me wipe out my annotations. So TLDR is take our assembly, feed it through all these different tools that try to use these different molecular biology properties that we've talked about to try to predict what these things do. If you're curious about BLAST and Hammer specifically, these are two chapters from Pavel Pevsner's textbook. So if you take CSE 181, you're actually going to learn about the techniques that these two tools use. BLAST does sequence alignment. Hammer uses these hidden Markov models. Both of them are super cool. It's a cool textbook. But yeah. So for our purposes, we're not running all of Proca. I forgot why that was. I remember I ran into issues trying to like write graders to grade it or something like that. But we are running Prodigal just to kind of give you an idea of what's going on. Prodigal is one of the many tools that are in Proca. But all of those are their own individual tools that Proca is just kind of chaining together. And yeah, this is prokaryotic gene recognition and translation initiation site identification. But basically, they have it specifically for prokaryotic gene recognition. You can have other versions that are for other stuff. Okay, so what do we get at the end? So let's imagine that I've run a tool like Proca and it outputs a genome annotation, an annotation of what are the genes here, what do I think they do, what do I think they are, etc. So here, this is a tool called IGV, the Integrated Genomics Viewer, and you can actually give it different genome annotations. And here, on this top part, this is the actual SARS-CoV-2 like curated annotation that virologists have crafted over the years. And this bottom one is an annotation that Proca output just purely based on the genome assembly. So taking the raw sequences, running spades, taking the assembly, running Proca, and then just spitting that out. This is the annotation that we get. And as you can see, it actually looks pretty darn good. So over here, we see this one, and these labels, Proca itself predicted based on other proteins it's seen in the past, probably largely based on SARS-1. But here we see like it has this 1A gene, which matches pretty close to this ORF1AB gene in the SARS-CoV-2 genome. It has this rep sequence that doesn't really match the existing annotation, so that kind of, in reality, falls within this ORF1AB gene. But then this one, S. So S is the spike protein. This is a super important protein for how SARS-CoV-2 functions. And it actually like perfectly predicted that. ORF3A, it perfectly predicted. It missed this E gene that I don't really know what it does. This M it got perfectly. This ORF6 it got. ORF7A it got. It missed ORF7B. And then this N protein it got. This ORF8 it got. These other ORFs at the end, 9 and 10, it missed. But it did pretty good. So why? Oh yeah, go for it. Go for it.
Can you explain a little bit about how this is laid out? Is the bottom the entire assembly like in a row, or is it cutting sections of the assembly and matching them up where it's going to match with nothing?
Ooh, great question. The question was, can I explain what is actually going on here? So the assembly itself, we're not showing the sequence, but you can imagine the assembly is this top line. So this top line, so the SARS-CoV-2 genome is 29,903 letters long. First position, last position. These are just kind of the positions laid out. And each of these different little windows is just one gene. So just defined as start position of the gene, end position of the gene, name of the gene. I don't know why the official annotation has multiple tracks. I think maybe they were created by different labs, maybe, or I don't really know why they have different tracks, but just think the union of all of these different little, like each of these small labeled start end label things is one gene. And then this is all the start end label genes that we predicted, if that kind of makes sense. And then we could then look at what is the actual like ACGT blah 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 sequence that makes up that by just slicing the substring of the genome assembly.
How do you say it?
Oh, so the question was, why did I say in the middle it skips it? So here, so this is the one that we created. And our Proca annotation was not able to predict that there was a gene in this small little window right here. So this white gap just means nothing is here. The only things that are like gene predictions are the blue blocks. So over here, it didn't think that there was any gene. Over here, it didn't think that there was any gene. Over here, it didn't think that there was any gene. And then over here, it didn't think that there was any gene. But like in the actual annotations, we do know of some gene sequences that show up in those positions. So it failed to predict those specific things. Yeah, great questions. Awesome. Okay, so why does this matter? Well, again, I want to emphasize this takes zero virology background. None of you are virologists, but as you're going to learn on Lab 2, if you haven't already, you can run these tools. So you, without any virology expertise, can come up with an annotation that's almost perfect. And when we're trying to like fight some novel pathogen, if a virologist has to figure out all this crap on their own, that's going to be really hard. But if you as the bioinformatician can come in and take all of this existing knowledge that you have, guess something that's not perfect, but is pretty good, it's much easier to then hand this off to the virologist. Whoops. Okay, kind of like, it's hard to like draw straight lines. You can then give your predictions to the virologist, and it's much easier for them to refine it than it is for them to come up with something from scratch. Does that kind of make sense? Yeah. Oh, did you have a question or just stretching? Oh, yeah, for sure. Okay. So we have like 5 minutes left. This was my last slide. I don't know if folks want me to like do stuff from the command line or if folks have general questions or what is most helpful for everybody. Or we could call it 5 minutes early if people want to like stretch their legs before lab or any questions. Okay, cool. Let's go ahead and call it here then. Thanks so much, folks. So Lab 2 is out. Again, as always, it's going to be due Sunday at 10 p.m. You can only get help debugging your labs in the lab section, so please come. And yeah, I will see you all on Monday. Bye, everybody. Any questions from folks on Zoom before I end the Zoom call? Feel free to type in the chat. Feel free to raise your hand. Yeah, no problem. Cool. Thanks, folks. Have a good one.